Where's Bobby? Huh? Missing Bobby?
No, no idea. We can't make
Where's Bobby?
Comes to back.
Bobby! Rufito, have you seen your brother? Bobby! 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 Bobby!
In order to correct the problems created by synthetic fertilizers, we can attempt to rehabilitate our soil and enhance its health by inoculating it with beneficial microorganisms. What is actively aerated compost tea? Actively aerated compost tea is neither conventional compost tea, nor manure tea, nor liquid fertilizer. All of these are produced through fermentation, by soaking compost, manure, or organic matter in water for a number of days or weeks, with the result that we will end up with an anaerobic mixture adding anaerobic pathogens to our soil. Actively aerated compost tea, on the other hand, if made properly, is compost tea obtained through a process of aeration, with the result that we will obtain an aerobic mixture containing a concentration of aerobic beneficial microorganisms. How does this process work? We physically separate microorganisms from compost with air bubbles. Microorganisms stick to the organic matter in the compost with biological glues. They can therefore not merely be washed off with water, but they can be knocked off into the water by a constant barrage of air bubbles through the use of aerators. Once the microorganisms have been knocked off into the tea, they should be kept alive and allowed to grow and multiply. We therefore feed them with molasses or muscovado sugar, fish emulsion, or seaweed. And we provide them with a favorable environment, that is, adequate oxygen, also provided by the air bubbles, and the right temperature. But how do we in practice brew our actively aerated compost tea? For a practical guide on how to do this, we refer to our video in YouTube under Organic Farming in the Philippines. A first microorganism that we can cultivate ourselves is lactobacilli or lactic acid bacteria. Did you know that lactic acid bacteria arrest full odors associated with anaerobic decomposition? The bacteria thrive on ammonia that is released in the decomposition process associated with full odor. Through their biochemical action, full odors are eliminated. Lactic acid bacteria are therefore used in organic pig and chicken raising and in aquaculture. Lactic acid bacteria are used as diluted spray on plants and soil. They promote plant growth, plant health, and soil life. Lactic acid bacteria are also used in composting. They help in decomposing organic materials and make nutrients available to our plants. Lactic acid bacteria produce enzymes and natural antibiotics. These in turn promote good digestion and nutrient absorption in animals and people. And finally, lactic acid bacteria prevents bacterial infections like, for instance, Salmonella, which causes typhoid fever and food poisoning, and Escherichia coli, which causes diarrhea, food poisoning, and UTI. How do we collect lactic acid bacteria? We fill 1/4 of a large enough container with rice wash. We leave at least 50 to 70% of the container free for air. We cover our container with a piece of cloth so that air can move in and out, and we store everything in a quiet, cool place that is not exposed to sunlight. After 5 to 7 days, all kinds of bacteria, including lactic acid bacteria, will have gathered in the rice wash. In the process, a thin layer of rice bran with a sour smell will have formed on top of the liquid. We remove the bran and transfer 10 centiliters of the remaining bacteria-infected liquid to another container. To this 10 centiliters of bacteria-infected liquid, we add 1 liter of fresh milk and cover it again. By adding fresh milk, we eliminate all the bacteria in the liquid except for the pure lactic acid bacteria. They are the only microorganisms that can survive in milk. And that also explains the lacto in their name. Lacto is a term associated with milk. After another 5 to 7 days, our mixture of milk and contaminated liquid has turned into a yellow liquid serum with a white-colored cake floating on top of it. We remove the cake because we only need the serum, which contains the lactic acid bacteria. How do we use this lactobacilli serum? We add a few tablespoons of our pure lactoculture to a gallon of pure water. Make sure, however, that the water is chemical-free. Chemicals, including chlorine, will kill our lactic acid bacteria. This mixture we can now use. We can spray it on our plants to strengthen the phytosphere microbes and thus fortify our plants. We can water our soil with it to promote healthy soil life. Or we can use it in our compost to hasten the decomposition process. A second set of microorganisms we can cultivate are beneficial microorganisms from the forest. Because of their biodiversity, forests are a sanctuary for a wide variety of beneficial microorganisms. We can trap these microorganisms and so collect them. In case we are far away from the forest, we can create a forest-like environment by collecting leaves and making them into leaf mold. How do we collect these microorganisms? We gather forest litter from underneath the forest floor surface or from the bottom of our leaf mold pile. Back home, we prepare a 2-inch high plastic container halfway filled with cooked rice. Cooked rice is rich in carbohydrates that attract microorganisms. We cover our container with metal chicken wire. These will keep the rats out, yet allow air to come in. We bury the container under the forest litter or leaf mold. These should be in a dark, humid place imitating the forest climate. After a couple of days, we retrieve our container. The rice is covered with molds of different colors, a sign of contamination by microorganisms. We add molasses by as much as 1/3 of the contaminated rice. We again cover the container, but now with a piece of cloth. After a week, our mixture will have become sticky and semi-liquid. We dilute the semi-liquid mixture by adding an amount of water equal to 20 times the volume of the semi-liquid mixture. Finally, we strain the whole thing. The resulting liquid we call our basic forest microorganisms. Another set of microorganisms are the bamboo beneficial microorganisms. These organisms are attracted to the sweet substance secreted by the roots of the bamboo. How do we collect these microorganisms? Well, this is done the same way as collecting our forest microorganisms. However, if we have bamboo growing close by, we can as well bury our container with cooked rice right at the foot of that bamboo. Our fourth set are beneficial microorganisms associated with specific plants. Each plant species has its own set of beneficial microorganisms specifically associated with it. The presence of these microorganisms enhances the health of the plant and optimizes its chances for survival. One well-known example is the Rhizobium bacterium on leguminous plants. So, when cultivating a certain crop, we can enhance its general well-being by first inoculating the bed on which it is going to grow with collected, readily available microorganisms specific to it. How do we collect these plant-specific microorganisms? We select a healthy, vigorous specimen of the crop we want to grow. We cut the plant and cover the cut plant with a plastic container of cooked rice. We do this in such a way that the opening of the container is facing the spot where the plant was cut. The beneficial microorganisms present in the plant will be attracted to the cooked rice. For the remaining steps, we follow the same procedure as in collecting forest microorganisms. The fifth set of microorganisms is Rhizobium. Rhizobium is one of the most common nitrogen-fixing bacteria present in nature. It has the capacity to take nitrogen from the air and to store or fix it in nodules on the roots of leguminous plants. This very important beneficial bacterium can also be cultured and added to our soil. After all, the more beneficial microorganisms in the soil, the merrier. How do we grow a Rhizobium colony? We pull out a bunch of leguminous plants while they are in flower or fruiting, because that's when they have plenty of nitrogen nodules on their roots. A very vigorous leguminous plant for this is paayap. We collect the soil right there where the plants were growing. That soil is pregnant with Rhizobium. We mix the collected soil with about the same amount of crude sugar. The Rhizobium will feed on that crude sugar and multiply. This mixture of soil and crude sugar in which the Rhizobium bacteria live, we can add to our leguminous seeds for inoculation. EM is the name we give to a collection of different beneficial microorganisms that we put together into one mixture. It is up to us what beneficial microorganisms we put into our collection. In other words, when creating EM, we put all our resources and beneficial microorganisms together. The more diverse, the more viable and potent our mixture will be. One important rule: 50% or half of our EM mixture should be lactobacilli. The other half is an assortment of beneficial microorganisms. For example, 1 part bamboo microorganisms plus 1 part forest microorganisms plus 1 part plant-specific microorganisms, such as, for instance, broccoli. To these 3 parts, we add 3 parts lactobacilli. Apart from carbohydrates, which we find in cooked rice and crude sugar, we can feed our microorganisms with yet other nutrients that will make them even more effective. For instance, fermented plant extracts, fermented fruit juices, fish amino acids, etc.